This is screen capture 8.2 of cellular respiration, and what I'm showing here on the screen is a cross-section illustration of a mitochondrion. And you can see the outer membrane, the inner membrane, crista here. And it's showing the chemicals that go in, and the other parts I should mention would be the watery inner portion of the mitochondrion called the matrix. Glycolysis occurs outside the mitochondrion in the cytosol, the watery portion of the cell. Pyruvates or pyruvic acids are produced. Those are moved into the mitochondrion, and there's a transition reaction or a preparatory step that occurs where it's converted to acetyl coenzyme. Each pyruvate is converted to acetyl coenzyme A. So 2 pyruvates, 2 acetyl coenzyme A's. Each acetyl coenzyme A eventually goes into what's called the citric acid cycle, and the carbohydrate, which began as glucose out here in the cytoplasm, is completely oxidized. Carbon dioxide is given off. That's shown here and here. And NADHs and FADHs are produced, a little bit of ATP, and that goes to run the electron transport chain, which occurs across the cristae, inner membrane. Okay, so this is glycolysis. Just to go back here and take a look. Yeah, it looks like I have it twice. I've mentioned that already. 2 ATPs are produced, 2 NADHs. So here's a short form of what happens in glycolysis. You start with a 6-carbon glucose molecule, and we keep track of those atoms. That's important with the laws of thermodynamics. If you were to take a chemistry class, a physical science class, physics class, but especially chemistry, that's one of the things you do. Make sure that your equations are balanced. You want to make sure that you keep track of the atoms. They don't just appear and disappear. So 6 carbons on the left, we end up with 6 on the right. The pyruvic acid, or pyruvate as it is sometimes called, is a 3-carbon molecule. You have 2 of those. It's a total of 6. 2 ATPs are produced, 2 NADHs. That's the short of it. A little bit longer, you start with glucose. There are a number of steps and intermediate products, and eventually you end up with pyruvic acid. And of course, what you're seeing there, it's happening twice, so you get 2 molecules of this pyruvic acid, 2 ATPs, 2 NADHs. These pyruvates, or pyruvic acids, are moved into the mitochondrion. In the mitochondrion, a transition reaction occurs. So where does it occur? In the mitochondrion. You want to know that. Each pyruvate is broken down into acetyl coenzyme A, and by being broken down, a little bit of carbon is pulled off. When those carbons are pulled off, and the molecules just rearranged just a little bit, 2 NADHs are produced, and of course the carbon dioxide is released from those carbons being pulled off. In this schematic, the short of it is you have a pyruvic acid entering. It's oxidized to acetyl coenzyme A, so it goes from a 3-carbon to a 2-carbon. Where's that 3rd carbon? It helps form carbon dioxide, and that's exhaled eventually. And an NADH is produced for every pyruvic acid. Since there are 2 of these molecules, pyruvic acids that go in, you're going to get 2 NADHs, and you're going to get 2 acetyl coenzyme A's and 2 carbon dioxides. The acetyl coenzyme A is in the mitochondrion, in the matrix. It goes into a set of reactions called the Krebs cycle. Sometimes it's called the citric acid cycle, sometimes it's called the tricarbolic acid cycle. It has 3 different names, but the text we used has stuck with the Krebs cycle, named after Dr. Krebs, who studied cellular respiration. This occurs in the mitochondrion. This is where most of the carbon dioxides are produced. I don't ask you to know how many, but most of the carbon dioxide is produced here. 2 ATPs are produced, 6 NADHs, that's the most NADH from any vocation thus far, and 2 FADHs. And the NADH stands for nicotine adenine dinucleotide. The FADH stands for flavonoidine dinucleotide. FADH contains just a little bit less energy than NADH. And so This is where most of the NADHs are produced in the Krebs cycle. This is where all the FADHs are produced. And the ATPs that are produced here are from substrate level phosphorylation. It's from the complete oxidation of what's left from the original glucose molecule. And the carbons that are pulled off go into carbon dioxide. The short of it is, acetyl coenzyme goes in and joins with another chemical. A carbon's pulled off in the form of carbon dioxide. Eventually, 2 of those will be, because we have 2 acetyl coenzymes going in. 3 NADHs. Actually, I say 2. It will be a total of 4 eventually. 3 NADHs for every acetyl coenzyme A that enters, an ATP for every acetyl coenzyme A, and an FADH. Since there are 2 acetyl coenzyme A's entering, we're going to have 4 carbon dioxides, 6 NADHs, a couple of ATPs, and 2 NADHs. And again, I'm not concerned that you know how many carbon dioxides. Showing it a little bit longer, for every acetyl coenzyme A that goes in, it goes around and there's some intermediates. Here's the citric acid. There's a alpha-ketogluteric acid, there's a succinic acid, a malic acid, and oxaloacetic acid as intermediates, which are further oxidized and energy is released in the process. And you don't need to know those intermediates. All the NADHs and FADHs produced thus far, and all the reactions I've mentioned, eventually go into what's called the electron transport chain or electron transport system. I've heard both expressions. This occurs across the inner membrane of the mitochondria, and again with those cristae, those infoldings of the inner mitochondrial membrane, that increases the surface area and allows for greater amounts of chemical reactions to occur, such as the electron transport chain. This is where most of the ATP is produced. Oxygen will serve as the final electron acceptor, and water is produced as a result. For every NADH that enters this electron transport chain, when it's oxidized fully, there's enough energy that will be able to be used to produce 3 ATPs. FADHs enter a little bit lower on this chemical reaction chain, and therefore they don't have as much energy. For every FADH that's broken down, enough energy will be released to produce 2 ATPs. Since these ATPs are not being produced from the direct breakdown of the original substrate, but they're being produced because of the flow of electrons, and hence hydrogens, across the membrane, the inner membrane of the mitochondrion, it's a chemiosmotic process, and therefore it's called chemiosmotic phosphorylation. Anytime you see this expression, chemiosmotic phosphorylation, link it to a membrane. It's occurring across a membrane. Here's a short schematic. NADHs enter, FADHs would enter just a little bit lower. Those are oxidized, and notice it's losing a hydrogen, losing an electron, or actually a couple electrons. This passes from chemical to chemical to chemical. These electron carriers that are designated in the blue, though they're not necessarily named, many of them are cytochromes. There's cytochrome A, cytochrome B, C, and so on. And so the electrons are transferred. And as the electrons are transferred, energy is given up and used to put ATP molecules together. Phosphorylation event. And it's chemiosmotic phosphorylation. The electron gets to the last electron carrier, and then it's passed to oxygen. The oxygen molecule is broken up. 2 hydrogens, 2 electrons are added, and you get water. And water is a result, one of the products of the electron transport chain, as well as most of the ATPs that are produced in the cell. When aerobic respiration is complete, that means the glucose molecule has been completely oxidized, and the NADHs and FADHs that were formed have been broken down in the electron transport chain. There can be as many as 38 ATPs in bacteria. Bacteria are a little bit more efficient at producing ATP than eukaryotic cells. In eukaryotic cells, the maximum ATPs will usually be 36. And that has to do, the difference is, because there's a mitochondrion in eukaryotic cells and not in bacterial cells. it takes a little bit of energy to get the pyruvic acid or pyruvates into the mitochondrion back from glycolysis before the transition reaction. And that amounts to about 2 ATPs, and that's why there's not as much ATP produced in eukaryotic cells. If there's no oxygen present, or low oxygen conditions, then some cells are able to undergo what's called fermentation. And this is under anaerobic conditions, but it's not anaerobic respiration. With fermentation, no oxygen is required, and it does occur out in the cytosol. No special organelles are required. The short of it is this. The glucose molecule is broken down in glycolysis, same as the beginning for aerobic respiration. 2 ATPs are produced, a couple of NADHs, a couple of pyruvic acids, or pyruvates are produced, and those continue in the regular fermentation reactions, and there are different fermentation reactions. In some fermentation reactions, carbon dioxide will be produced, and you're going to get some kind of an organic product that still has energy to it. Here they're showing ethanol. It could be lactic acid, but here they're showing ethanol, and there's still energy in this, and it is possible to use ethanol as an energy source. Usually, the products of fermentation are toxic to the cells that are producing them. We don't produce ethanol, we produce lactic acid. And lactic acid will quickly make our muscles sore as it builds up. Our liver breaks it down and processes it back to a usable form, but our body doesn't allow it to build up to where it's toxic. For cells that are producing ethanol, such as yeast cells, if they are allowed to continue producing the ethanol, they'll actually kill themselves. They'll pickle themselves in their own ethanol. So if you're in alcohol production, you have to remove the alcohol or remove the cells so they're not all killed off by the ethanol production. In this list down below, I have products of fermentation such as lactic acid, that's what we produce, ethanol, yeast cells, some bacteria produce it, but think of yeast cells, which are a type of fungus, produce it, and they produce ethanol when oxygen concentrations are low. If you're trying to produce ethanol and you provide plenty of oxygen, generally that won't happen. The yeast will undergo aerobic respiration because they are facultative anaerobes. They can do either aerobic respiration or fermentation, depending on oxygen conditions. Formic acid, ants will produce formic acid. Butyric acid, that's found in vomit, makes vomit smell really bad. That's one of the reasons why you'll use a base to try to neutralize it and control the odor. Acetic acid is found in vinegar, so when you step into a kitchen where somebody's doing pickling, they're using vinegar, you're smelling the acetic acid. It was also used in film development, in photography. Acetone is an organic byproduct, has different uses, especially to dissolve organic substances. Citric acid is used as an additive to many foods and drinks, especially soft drinks. Methane is a CH4 is the chemical formula. It's a single carbon molecule that you can burn. People burn it with natural gas. That is what natural gas is made of. Carbon dioxide and hydrogen gas are other byproducts of fermentation. That brings us pretty much to the end. I will have some thinking questions for you. You can hopefully work through those, and if you happen to have any questions, be sure and let me know, and I'll try to help you out. Hope you do well on the test.